Okay. Um, Hello everyone. Welcome to today's new Zoom seminar. Um, it's One thing before the seminar, I want to make an announcement for everyone online. So in US, from this week on, we're gonna switch to the winter time. So if any audience from the US, we're gonna have 1 hour earlier. So it's if it's 9 a.m. in Beijing time, it's 8 p.m. in East time. Just to keep in mind. So really my pleasure to welcome our today's speaker, Dr. Zhihua Gao from Zhejiang University. Zhihua currently is a professor in Zhejiang University Brain Science and Brain Medicine Institute. Zhihua finished her bachelor degree in Xiangya School of Medicine, Central South University in China, and then continued the master's degree also in Xiangya School of Medicine. So after that, she decided to pursue her PhD in Canada, complete her PhD training in University of Alberta in Canada. So and then after a short period postdoc training, she moved to Department of Pathology and Immunology in Baylor College of Medicine in Houston, and returned back to China from 2013 as a associate professor in Neuroscience Institute in Zhejiang University School of Medicine. So from 2013 until today, she did a remarkable job and then get promoted as full professor, still in the Neuroscience Institute in Zhejiang University. So Zhihua's lab is mainly focused from the regulation of cell metabolism, and in particular interested in the microglia biology. They actually focus on the role of cell metabolic reprogramming in microglia activation, and then using different approaches, including real-time observations of the impact of metabolic manipulation on microglia activation through intravital microscope imaging technology, and combined with other methods such as different omics analysis, flow cytometry, and also transcriptome analysis to look for effective target which can be manipulated and analyzed the role of cell metabolism in microglia mediate immunological disease. And at the same time, they also are interested in studying the brain development and related disease to provide a relatively new perspective for understanding and intervening in the accordingly disorders. So her research team actually put a lot of effort to understand the endocrine neurons in regulating social interaction and emotions. So her lab not only focusing on the microglia, also in the neuroscience, she did quite amazing progress in the field. And then with that, to map anatomic and then molecular connections, as again using different approaches to explore endocrine neural activities and their roles in regulating body behavior and homeostasis and their connections with the development of effective diseases. I think from Zhihua's publication, you appreciate her really put a lot of effort to promote microglia biology, both from the angle of immunology and the neuroscience. So I think today, Zhihua probably gonna give us some update of her latest research progress in the lab, again, related to the microglia metabolism. So with that, thank you very much, Zhihua, to present your work, and then we are very much looking forward to it. Please go ahead.
Thank you very much, Chuan, for the nice introduction. It's way beyond my thoughts. Thank you. So good morning and good evening, everyone. Many thanks for Chuan's invitation, and particularly thank you for the continuous efforts to carry on this immune Zoom seminars over these years. So I'm Zhihua, as Chuan already introduced, and my life. Okay. So, okay. My lab has been focusing on the homeostatic regulation in the brain, as Chuan already introduced. We are trying to understand how immune and neuroendocrine interactions coordinatively contribute to brain function and behaviors. So for today's talk, I'm going to focus on our recent findings regarding how micrometabolism, and particularly a key metabolic regulator, how it shapes microglia function. So as we all know, microglia are the primary immune cells in the brain and different from other immune cells in peripheral organs. Microglia are originated at a very early stage in the embryonic phase. They are originated from yolk sac primitive macrophages and then migrate into the brain at a very early embryonic stage. And then later on, they started to expand and colonize the brain parenchyma as well. They adopt different morphology, such as in ramified states or amoeboid or hypertrophic morphologies in disease states. So with this, different states or their rapid responses, microglia are very important players in regulating brain function. And like I mentioned, because microglia, they enter the brain early, they govern the brain development, and they rapidly associate with different elements in the brain. For example, at the early developmental stage, microglia closely associated with neuroprogenitor and the precursor cell. They help to regulate neurogenesis. During synaptic development, they facilitate the remodeling of synapse pruning. As well, they closely associate with vascular or oligodendrocyte to promote melanogenesis and regulate vascular genesis. So under disease stage, microglia elicit inflammation to modify the disease. So overall, they are actively engaged in a wide range of activity in the brain under both physiological and pathological conditions. Microglia, they are very busy. They never stop monitoring as a sentinel. And even in the healthy brain, they extend their ramified processes to carefully monitor the local microenvironment. Once there is an injury in the brain, they quickly respond with their processes and go directly towards the injury to wrap the injury and help to promote the tissue repair. So they, microglia, they continue to survey the parenchyma. They quickly respond to the damage. They also phagocytose invading pathogens. As well, they elicit rapid immune responses. So these amazing features make us always wonder, are there any particular program in microglia that metabolically support this business of microglia? To address this question, we compared the expression of 1,000 metabolic regulators in the brain using the already available public source as shown here. So we examined the expression of this metabolic genes from different sources, and what we found is that microglia indeed express a distinct metabolic clusters. As well, if we carry out the principal component analysis, we'll be able to see microglia does have a distinct expression profiles of those regulates, which is quite different from other brain cell types, such as astrocytes, neurons. Even in the most famous metabolic pathways, such as glycolysis or gluconeogenesis, microglia already showed a clear separation from other brain cell types, suggesting microglia do harbor distinct metabolic regulators that may help to support their uniqueness in the brain. So we then further compared those differentially expressed genes together with those 4 major cell types in the brains, including neuron, astrocytes, oligodendrocytes, and microglia. As shown here, we enriched a cluster of 25 differentially expressed genes in microglia, with 17 of them are highly enriched in microglia, while as 8 of them is significantly lower in microglia compared to other cell types. So among those 17 enriched metabolic regulators, HK2, the hexokinase 2, caught our attention because hexokinase, as we already learned from the biochemical textbook, is the key enzyme that drives the phosphorylation of glucose during the first step of glucose metabolism. So a little bit knowledge about the hexokinase. Hexokinase drives the phosphorylation of glucose into glucose 6-phosphate. In our body, there are actually 4 major hexokinase isozymes. So from hexokinase 1 to hexokinase 4, these hexokinases have been shown to express in different tissues. For example, in the brain, previous studies have already shown that the main hexokinase isozyme is actually HK1, which is quite different from what we found HK2 is enriched in microglia. While as the HK2 has been shown to highly express in skeletal muscle, cardiac muscle, which is responsive to insulin. And hexokinase 4 is the glucose kinase that we learned from the biochemical textbook. So while there is another hexokinase 3, which is very rarely studied. So among these 4 hexokinases, hexokinase 1 and 2 are the enzymes that with the mitochondrial localizing domain, they are localized at the outer membrane of mitochondria. And among these 4 members, hexokinase 2 is unique in that it harbors 2 catalytic domains, whereas other isozymes only have 1 catalytic domain. And this makes hexokinase 2 the most active isozymes among these 4 members. While they all drive the phosphorylation of glucose. That's the only step. So you may wonder, why do we need so many isozymes? It's because by phosphorylating glucose into glucose 6-phosphate, at this point, it's a very critical divergence point for the glucose metabolism. Where does it go? Really depends on which enzyme catalyzes, because these enzymes can bring their partners to drive the downstream pathway. And as we know, glucose 6-phosphate can either go pentose phosphate pathway for anabolism, or they undergo glycogenesis for glycogen storation. So if they go further to produce the phosphate biphosphate glucose biphosphate, and then undergo the anaerobic glycolysis or oxidative phosphorylation to produce energy, that will be the major catabolic pathways for glucose utilization. And studies have shown hexokinase 1 and 2 in the brain and skeletal muscle. They are mainly used for the catabolism that has been used to either generate a large amount of ATP using oxidative phosphorylation, or for very rapid ATP production through glycolysis in the skeletal muscle. However, I'd like to draw your attention is that hexokinase 2 has been recently shown highly enriched in tumor cells. And this high abundance of hexokinase 2 helps to drive the pentose phosphate pathway to produce ribophosphate and NADPH. That is mainly for the anabolism in the tumor cells, promoting tumor cell growth. So different hexokinase isoforms have different distribution, and they guide different phases of glucose. So as I mentioned, by data mining, we found HK2 might be selectively enriched in microglia. We then used both in situ hybridization and Western blots to further verify that HK2 is specifically expressed in microglia in the brain, well as shown here in the culture system, both astrocytes and the neurons predominantly express HK1, which is consistent with previous findings showing HK1 is the predominant hexokinase in the brain. So the question is why these cells need such different types of hexokinase. Before we ask that question, we further and this particular pattern of HK2 has also been shown by Zhejiang's group from Zhejiang University. So this has been verified. As a further verification, we generated HK2 promoter-driven TD tomato reporter line, and shown here. So the TD tomato fluorescence is completely overlapped with IBA1 staining, which shows particularly the microglia distribution, but there's no overlap with neuron or oligodendrocytes as well as astrocytes. So further verified, HK2 in the brain is selectively expressed in microglia. So then what's the function of HK2 in microglia? Does it, as for our initial aim and interest, we would like to see whether there are particular metabolic determinants to regulate microglial motility, and are they also, is this enzyme also involved in microglial homeostasis as well as their immune responses under different disease states? So to answer this question, we first did a straightforward experiments by deleting HK2 in the primary microglia culture. So with this, we made this mice and carried out the tamoxifen injection at postnatal day 1 to day 2, and then perform this mixed culture and collect primary microglia. So this has already shown a very effective knockdown of HK2 in the cells. We then carried out the seahorse assay to measure the glycolysis and oxygen consumption rate in the cells. As shown here, the deletion of HK2 has significantly reduced the rate of extracellular acidification rate, which suggests without HK2, the glycolytic activity of microglia cells is significantly reduced. As well, the oxygen consumption rate of the cells, particularly at the maximum stage, is also reduced. So those were carried out in the murine microglia. We then further verified this by isolating microglia from a human brain and then inhibit the activity of hexokinase 2 by the 3-bromopyruvate. So this is which is an inhibitor. As well, we also observed reduced glycolytic and oxidative phosphorylation activity in human microglia. And together, this data showed that HK2 deletion reduces both the oxidative phosphorylation and the glycolysis, which reduces the ATP production in microglia cells. So how does this affect microglia surveillance? Using a 2-photon microscopy imaging, what we first observed the basal surveillance of microglia, as shown here, within a 20-minute window. We did see attenuation of the microglia's basal surveillance, as indicated by the process extension and retraction. So there is a difference, but small. However, when we introduced an injury by the laser within the brain parenchyma, we will be able to see that microglia cells without HK2 showed significantly slower migration towards the injury. So so by half an hour, by 30 minutes, usually their wild type counterparts are getting close to the injury and a fully wrapped injury, whereas in the absence of HK2, their processes are still halfway on their route, migrating towards the injury. So with quantitative analysis, we'll be able to see that their speed and is significantly reduced, presumably due to a reduced energy production efficiency. So we've showed you that HK2 deletion affects basal and chemotactic activity of microglia. How does it affect microglia homeostasis? And unfortunately, when we knock down this in the adult brain, we carefully observed the number, density, and the morphology of microglia. Unfortunately, we didn't see any differences in these cells, no matter regardless of the density, morphology, and number. So that was to our great disappointment. So we then thought about, so maybe at the because usually at within the intact brain, the turnover rate of microglia is very low, and that's probably why when we remove HK2, doesn't really affect the homeostasis. However, if we introduce a challenge by quickly depleting microglia using the colony's stimulating factor 1 receptor inhibitor, PLX5622, like after 2 weeks, all the microglia are killed. And then we put back the normal diet without inhibitor. In this case, usually within 1 week, microglia can quickly repopulate. And then during this period, we were able to see a big difference during the repopulation of microglia phase. For example, at day 1 and at day 3, the wild-type microglia quickly repopulating with high proliferative activity. However, microglia without HK2 showed a particularly slower, dramatically slower proliferative activity. So suggesting that there is a delay in microglia repopulation. However, this delay was caught up at later stage, and that microglia quickly recover. And we don't know how they recovered yet. So this delayed repopulation of microglia in the absence of HK2 is due to a transient cell cycle arrest, primarily in G1S transition. And so then after examining how it affects microglia motility and microglia homeostasis as well as their repopulation, we then really want to ask questions how this metabolic feature contribute to the immune response of microglia. As we know that immune cells undergo rapid metabolic reprogramming during to elicit effective immune response. For example, LPS can quickly promote aerobic glycolysis, while as inhibiting oxidative phosphorylation to facilitate inflammatory responses. And previously, we've already shown that we've also verified this in primary microglia that LPS can promote this dramatic aerobic glycolysis and suppressing the oxidative phosphorylation. So with this, because when we knock out HK2, we observed reduced glycolysis. We thought, in this case, if we treat microglia cells with LPS, we would be able to see a reduction of the inflammatory responses. However, to our surprise again, when we remove HK2 in the microglia cells, and after we further challenge microglia cells with LPS, or poly-IC, a viral mimic, we observed dramatic elevation of inflammatory cytokines, including interleukin-6, TNF-alpha. So this suggests HK2 deletion didn't reduce inflammation, but instead it enhanced inflammatory response in microglia. So why do we have this contradictory data from what we observed, like from metabolic reprogramming before? So we further verified this effects by using a in vivo photo thrombic stroke model. In this case, so my student Fang did a inject the rose angle into the mouse, into the vessel, and then induce the thrombosis by laser. In that case, we will be able to see a stroked area in the brain, and then she extracted the tissue in the disease tissue out and compared the inflammatory cytokines levels in between wild type and HK2 knockout mice. Again, she also observed the elevated inflammatory responses in the absence of HK2. So suggesting indeed HK2 deletions enhances rather than reduces inflammatory responses in microglia. So how could this be? We then further, as a result of this increased inflammation, Fang also observed like exaggerated stroke, the damage within the brain. As shown here, there is enlarged ischemic core area, along with this impaired behavior. If we put the mice under the going cylinder or grid walk test, so they showed even worse behaviors. So why does HK2 deletion produce such a contradictory effect as we expected before? As I mentioned, so both HK1 and HK2 have mitochondrial localizing domains. They localize at the outer membrane of the mitochondria. At this outer membrane, they closely associate with the voltage-dependent ion channel, which together they comprises this mitochondrial transmembrane potential, the pore, which allows the flux in and out of different ions as well as ATPs. So we then thought, okay, maybe by deleting HK2, it actually, because previous studies also showed HK2 may be important for the integrity of the outer membrane of the mitochondria. So we then thought, okay, perhaps when we delete HK2, this integrity also get impaired. In that case, so if we examine the mitochondrial membrane potential, there may be a change. So as shown here, when we remove hexokinase 2, there is indeed a reduction of mitochondrial membrane potential in this microglia cells, suggesting it indeed disrupted the integrity of mitochondrial function. As well, we then further examined whether this interruption has further effects regarding the reactive oxygen species levels. So as shown here, removing HK2 in microglia actually increase the levels of mitochondria reactive oxygen species. And so if we use this particularly a mitotemple to scavenge this reactive oxygen species, we were able to reduce the enhanced inflammatory responses that were introduced by the removal of HK2. So suggesting this is actually, so HK2 can regulate mitochondrial function by harboring at the outer membrane and regulating the ROS levels. So to summarize this part, we propose that HK2 as a classic glycolytic enzyme, they actually play a dual role by first gating the glycolytic flux at the first step of glucose metabolism. Second, by localizing either outer membrane of mitochondria, it also regulates mitochondrial function through a non-metabolic way, which actually further regulate mitochondrial membrane potential and ROS levels. Through this, so that's why when we remove hexokinase 2, we observed enhanced immune responses in microglia cells. Through the metabolic pathway, microglia HK2 shapes microglia motility and their proliferation states. So, and this dual levels of hexokinase 2 activity helps us to understand what we've seen in different states of microglia, how removing HK2 affects microglia function. So what's really interesting is that we know that brain is a big glucose eater with 2% body mass killing using 25% of glucose. And previous studies have already all predominantly focusing on the usage of glucose by neuron and astrocytes. Here we've showed you that microglia actually express the highest active hexokinase 2 as well. And this is very important for its function, as recent studies are also showed compared to neurons and astrocytes, microglia actually exhibit the highest glucose uptake, even in the physiological stage, while as under pathological conditions, when microglia are active, their glucose uptaking capacity is even higher. And combined with the highest activity of HK2 within microglia, so the microglia are really the sweetest cells in the brain. And this sweetness is very important for microglia, as they are the minority in the brain. And in order for them to rapidly continuously survey the brain parenchyma and rapidly respond to invading pathogens and local injuries, microglia need to be armed with high glucose uptake and utilization capacity so that they can actively engage in diverse functions. So from that, so by saying that the sweetness of microglia is important to support its function, and we also observed that hexokinase 2 is very sensitive to microglia states. As I just mentioned, others already showed when microglia are active, their glucose uptaking activity increases. And here we also showed not only their glucose uptaking activity increase, but they also need a way to utilize the glucose, and the upregulation of hexokinase 2 will be a very good fit. So as shown here, after LPS treatment, HK2 goes up in AD associated microglia or other diseases. All this, when microglia are in reactive states, HK2 levels are elevated. As shown here, like in MPTP-induced Parkinson's disease models, HK2 levels are also increased. And Zhejiang's group also showed HK2 upregulation not only in Parkinson's disease model, but also in the patients. So as I showed, we've generated this TD tomato line, and this line actually can very vividly show the increased level of HK2 as shown here in this photostrophic stroke models. When microglia gets activated, the level of HK2 and reflected by the TD tomato fluorescence is also increased. So this increase actually correlate with the states of microglia, suggesting that HK2 can act as a metabolic sensor in microglia. And since abnormal glucose activity and metabolism have been shown to be related in different brain diseases, so we wonder, perhaps we can use HK2 as a good tracer to reflect the metabolic states of microglia and to track the metabolic activity of microglia in different disease states, likely similar to the TSPO PET imaging. So, and Dr. O'Neill have suggested that immunometabolite pathways can be a very good target to for anti-inflammatory disease treatment. We thought perhaps using HK2 as a good tracer, we may be able to track the microglia metabolic states and to indicate the inflammatory states in the brain. So with this, I'd like to thank my group members, particularly Yaling, Kelly, Fang, for their dedication and contribution in this microglia metabolism project. And our group members also have other projects that I apologize that they are not featured in this talk. And hopefully we are going to have more to present in the future. And thank you very much for your attention. Also, we have during the course of this project, we've had support from all of our collaborators, such as including Dr. Shuming Duan, Wu Ping, and Yi Chen, and our research is funded by different agencies. And thank you very much for their support and for your attention.